So just mix the methods and figure them as you go. It was sort of a topic that jumped on top of my head when we discussed with Claire about this. And what I just meant to say is I'm working on mixed methods, and this is what I'm applying them in my research. And majority of them are iterative, so that's why you see figure things as you go. But my work is primarily to map antimalarial drug resistance, and I will share more of that. So you'll hear much about the methods, malaria, and drug resistance. But I know we have people from different sort of specialization. So a little bit about malaria, it's still a global health burden. And you will see on the map on the right side, Africa still has majority of the cases, and areas of highest transmission, majority of them are in Africa. You'll see a little bit in Asia and the Americas, but majority of it is still occurring in Africa. And WHO prefers, and a lot of programs prefer, this type of classification, because even within a country, you'll still find this sort of the difference in the burden within a geographical area. And the highest threats now are drugs, insecticides, and actually diagnostic resistance. So what it means is the current drugs that we use for malaria, the parasite is getting more intelligent on the mechanisms we use to kill them. So they're trying to evolve and survive. It's a little bit easier to speak of resistance now and mutants, because actually what happens to the malaria parasite as well, as we have seen in COVID, it changes. And we have different variants in malaria as well, that are resistant to either drugs or insecticides or diagnostics. And these have been reported across all the regions, Africa, Asia, Americas. And so we will speak today about surveillance of antimalarial resistance. And as we know, WHO sets the stage and of the public health practice, evidence practice. So how do we do surveillance? So we use the therapeutic efficacy studies. These are the global standards, sort of the gold standard. And you will see also there are different recommendations depending on the transmission of malaria. So actually what happens with therapeutic efficacy studies is just you give a drug to patients, you follow them up, and different drugs have different follow-up periods. So some of them could be 21 days up to 48 days, depending on the type of malaria as well. I might be wrong. I don't remember the days correctly. But the aim is to see if someone had malaria, they got a treatment. First, you have to make sure that they take a treatment. So it's an observed treatment. And then you have to follow them up to see how they respond to the drug, which will also include to take some blood samples and measure the a little bit of the concentration of the drug, as well as to know the parasite density if it's getting low. And if it's not happening, then we can document that. And later on, either you can proceed to be able to evaluate if there are any molecular markers, if they have a variant type of malaria, or if it's just normal malaria. And we also, different individuals respond to the drugs and malaria differently. So therapeutic efficacy studies are the gold standard, but then other measures that are there are integrated drug efficacy. Now, the difference with therapeutic efficacy is just that in areas of low and very low transmission setting, you should know that this is more of a um, quantitative data um, analysis. Therefore, it's bound to sample size and the number of samples. But then with us trying to eliminate malaria, we're getting fewer and fewer cases. So there are places where they cannot reach a sample size. So what WHO recommends is to form sort of a therapeutic efficacy study using different health facilities. You'll see in therapeutic efficacy studies, most of places have sentinel sites, and sort of they monitor these maybe after every 2 years. But for places where they have low, some moderate, low, and very low transmission, they cannot achieve that sample size even after a long time, and it's an expensive activity. So therefore, they're trying to use integrated drug efficacy. And lately, probably not very late, but of the recent, uh, molecular markers have come to be the biggest player within this evaluation of drug resistance. And what happens here is, I'll give an example of Southeast Asia, for example, is in the different either sentinel sites or clinical trial sites. First, what they do is to do genotyping of the malaria parasite, and there's different ways of doing it. And there is for identifying if any of those variants that have been already identified can be seen within the parasite that is circulating. So, or it could be several malaria cases that have been treated, and um, eventually those that don't respond very well to the treatment end up getting this molecular evaluation. So it's being done in different ways, but it can act as an early warning system, but also it can be a surveillance in itself if integrated into the either routine treatment or within the routine surveillance system, even in sentinel sites. So that's what has been happening. So I've been saying this artemisinin resistance. I didn't include the definitions, but actually what we mean is just the um, we expect a patient to respond using a certain amount of drug in a certain amount of time. And a patient's response to the treatment is actually how they respond, they start feeling better, but also how the parasites within their body are getting cleared. So if the clearance of the parasite doesn't reach the optimum time that is expected, it's sort of prolonged, and actually there are even specific definitions for how long, then we start suspecting there is a problem with the efficacy of the drug or that patient responding to the treatment. So using these categories, at some point they started seeing a change on how patients respond to the drug. Taking you back to history, we have had different types of antimalarials. And back in early 1900s, we started using chloroquine, and later on, I think 20, 50 years later, after it was discovered, we started seeing resistance to chloroquine. So people would get treatment for a correct dosage, but they wouldn't respond. Then there were new antimalarials that came into play. We had sulfur drugs, but also they ended up getting resistance. So resistance in malaria hasn't been a new topic. But why is it important now, especially to artemisinin? So this is artemisinin and the combination therapy. So find artemisinin and another drugs that are combined are the main treatment now globally for malaria. And it hasn't been easy to find other antimalarials. And it was almost a magical drug, and a lot of papers were published in early 2000s when it came into play. And some countries, especially in Africa, started using it after 2004, 5, up to 2010 in different combinations. But in Southeast Asia, some of these drugs were used as herbal treatment. It's a common plant that actually grows in different places in the world, but it was sort of incorporated into treatment longer before, especially in Asia. And the first reports came from 2008 from different publications that this drug also was getting resistance. And in around 2013, 2014, there was some researchers worked to find what was the link of drug resistance, especially for the artemisinin drug, and they found that the parasite actually has changed the genome, and at a specific location, which is the K13, within the K13 gene, they can sort of be irresponsive to the drug. Now, from this, it gave scientists a window to be able to sort of measure, sort of to detect if a parasite that is being harbored by a patient is resistant. So this is what we had. So a lot of genomic studies started, and including there's a worldwide map of drug resistance that was done in 2014. This was published, and it incorporated samples from different parts of the world. The Worldwide Antimalarial Resistance Network started also mapping, coming up with a survey, and then until today, if we move to 2021, actually up to mid last year, reports were only coming from Southeast Asia, mostly, and 5 countries in Southeast Asia, in the Greater Mekong sub-region. And then later on, we had some few reports from India, which are still sort of questionable whether it's true, but molecular markers of drug resistance were detected there. And then early this year, late last year and early this year, there are more reports that are coming also from Africa. We should know that the parasite has already shown so there are 2 mechanisms. Either the parasite itself evolves intrinsically to develop this resistance, but also there are mechanisms, especially in Southeast Asia, where we have seen a certain type of parasite becoming more prevalent, and the more prevalent it becomes, the more drug resistance it causes, especially in the western part of the Greater Mekong sub-region, northwestern. And so, for example, for the reports that are coming from Africa, they could be independent emergence of this resistance and not necessarily transmission. So these are the things that are coming up into play. But so what we know so far, antimalarial drug resistance generally occurs in areas of moderate to low transmission, and that's what was happening. That if you clear the parasite load, sort of the ones that are remaining, like the last parasite remaining, are the most resistant ones. So those were the assumptions that were carrying on, and it is a threat to malaria elimination. But then who responds to the drug resistance? Just as we see in COVID today, despite we have variants, alpha, gamma, delta, it's on governments and policymakers to act on measures on how to reduce this spread and protect their people. Now, even for malaria as well, the policymakers actually are the ones in the front line. These are malaria programs people from the government and different international partners who also work in malaria. But then the challenge comes, do we always have to wait to do therapeutic efficacy studies despite knowing that they're very expensive and difficult to run. Actually, there are different WHO reports that show a lot of countries can't be able to even afford doing them. So one of the recommendations from the new the framework for malaria elimination, WHO recommends starting using routine surveillance as a potential way of monitoring drug resistance. But in reality, there's very little research that supports routine surveillance data can be used for that. And another thing that I can show you, so this was the worldwide map of drug resistance that came in 2014. You'll see a lot of countries are being marked using either one color. So for example, my lovely country, Tanzania, it's mapped with one color, same as different places in Brazil. So this one big dot, but exactly what does this one big dot mean? Does it mean if we zoom into Brazil to this area, is it all of that area having, let's say, this kind of mutations, assuming if these were the resistance mutations? Or is it just a little dot there, but because of they have zoomed out, then it looks bigger? Or the areas that are not marked in this map, does it mean they don't have any of this resistance? So this is the one surveyor, which also maps drug resistance. And you'll see it uses dots, which location sort of signs. And what they do here, actually, they map this where data was reported. So sometimes if a data wasn't study site wasn't reported, they use the country's capital city. And just to go back, in actual worldwide map published by the Institut Pasteur, here they were actually using, they also, in their data set, if you look, they were using the country's capital cities. So, and then a better map that came from them was, you'll see these boxes like C58Y, I543T. So these are the mutants, just like how we would say probably Delta, Gamma, Alpha of COVID-19. But then they are put into different geographical locations. So let's say this is Cambodia. They have put a group of them occurring here. And here they're saying, actually, this is the distribution of these haplotypes. But then in reality, if we are thinking of a map, which is a sort of geographical representation of a field on Earth, does that mean that this whole area in probably Laos or Cambodia, does it harbor those mutants? So in reality, you'll see different types of maps, and even if you open up any map today, there's no actually a consensus of best practice in epidemiological cartography. So the most recent guideline is from the CDC, which sort of puts guideline of epidemiological maps, but majority of that are borrowed from geography. That which will tell you, please, if you make a map, make sure the north arrow points so that people know the north arrow. You put the scale, you put the legend. So it's not a science that is universal so far that is known. And so this is if you zoom into the surveyor. So this gave rise to the PhD topic which I've been working on antimalarial drug resistance. So the important part was, if we want malaria policymakers to have visualizations, maps, or tools, what can we use, or what should we give them? And what kind of tools do they want, and how should we make them? So So this is actually the topic that I've been working on, and my aim is to enhance surveillance through improving the quality of anti-malaria resistance maps and their relevance to malaria programs and policymakers. So we wanted also to see how drug resistance in the Southern Africa side would compare to Southeast Asia, because um, when we're looking at malaria elimination, a lot of the Southern African countries have a low to very low transmission burden. So they are somehow relating to what's happening in Southeast Asia now. So if drug resistance was to become a problem, probably these would be the areas. So I'll put that in quotation, because now we are seeing it arising from different areas. So this was the aim back in 2018, and there's research gaps that we're trying to respond to. Where first, we don't know the suitability of using routine surveillance and secondary data for tracking drug resistance. And then we know that there are no consensus on epidemiological tools of how to map diseases, especially infectious diseases. And then how do we integrate the policymakers or malaria managers' specific recommendations into the kind of tools that we provide them or visualization that we make? So, but then the question would be, how do we make maps? Yes, we use some data, we use probably some software. Back in the days, people just sit down and draw using pencil and pen and paper. Uh, then there's someone who holds a pen, that's the cartographer. It's a scientist today who would be able to manipulate the software and then mix things together, probably this data and some colors, and then you get a map. But are all maps the same? And this makes it very easy now. Anybody can make maps, especially with the power of software that we get, but probably not almost everybody. Um, and when we are talking about maps, what do we know about them? So we know they are powerful, useful tools, but they may mislead. This guy wrote a good book. I would recommend, How to Lie with Maps. Maps have caused wars. People would choose to highlight things. Just a little bit of highlight or zooming in can make a problem look bigger. So it can over miss or and identify a problem. And then we are using data, but where does this data come from? What are the methods that we use? The kind of software we use, do they come with problems within them that can cause us to over miss or and identify the health problem? And this would be important because we're also using colors, dots, symbols, lines, and images to show maps. But some research has shown um, the primary colors one is used to, or the language. For example, I'm a native Swahili speaker. If in Swahili we can only say we only have names for 4 or 5 colors, if I receive a map that has 10 colors, would I be able to understand it? So some research has shown that actually uh, language perception within community, let's say black probably means bad, white means good in some contexts, but if you go to other cultures, black means good and it's worn in birth celebrations, and the white is worn in funerals. So do this change of how someone would look at a map and understand. Now, always if you see a map, you see red means bad and green means good, but is that a universal understanding? So these are the kind of things might make any map reader not understand a map just because they carry these things. So then map readers, they also have other challenges to understand or and sort of read a map. How do we learn from them? So we probably in the beginning we thought probably we need to learn about data, we need to learn about audiences, and then we need to learn about maps, and everything that makes mixed into the maps. Then to draw those maps, and probably we need to get a feedback and optimize that feedback. So that's how we landed into the mixed methods research. So um, we decided to settle with a sequential explanatory mixed methods design. At first, we wanted a meta-synthesis, a systematic review to sort of ground down the cartographic methods of infectious diseases. So the aim at first was, if we're going to make maps, let's first sit down and say how we're going to make these maps. But we can't just make this up. Let's look at the evidence. What does it say? And then we would use data. But in order to make a map, you would have to analyze this data quantitatively. So and wanted to see two types of data: published data and routine surveillance data. So what kind of maps can we get from routine surveillance data, from published resources? And how can we do the same using routine surveillance data? So we'll use spatial and the temporal descriptive. And then there are also, but we wanted to understand what so eventually we'd be able to have maps, but then we want to understand what are the the user's perspective, or how do they understand these maps. So the other way around would be to first go to the users, understand what kind of maps they would want to have for malaria elimination, especially with drug resistance, and then we would come and find the data that can produce such kind of maps. The data that can produce such maps, then the type of cartographic method we can use to produce the maps. But we thought maybe it's better we start with quantitative data, with what we have, and then, so this was more of convenient choice. And one of the most questions I receive is, why didn't you start with the other way around? But then how do you analyze, or how do you get people's views on the map? So we decided to use two methods. So we use semi-structured interviews, which is sort of, we're assessing usability. So if we create a map, then we identify what audiences we are trying to target, and then they would sit around the map, look at it, and tell us what they think. But then sometimes it's not only about the map, it's from the systems that produce data that created this map. So map are made from GPS coordinates, are made by cases of patients, for example, for routine data. So, and sometimes the feedback comes, no, this is not where we expect to see malaria. Why do you have it on the map? And that raises the question of data, or the software that we used, or the type of summarization we did. So we need to go back to the system and look at it and see where the problems are. And then the part of designing a map, making maps, we can use statistics to get our variables, but when you choose a dot or a type of shape or type of color, where to put it, how to put it, are you going to use a 2D map? Are you going to use an interactive map? This is a design process. It goes beyond statistical analysis to trying to understand what kind of visualization good to an audience or to your topic. So the design process, which includes map optimization, and sometimes it's not just a map, it has to be a tool where someone needs to filter and see, this is where I want to go, this is the type of information I don't want to. So these are kind of the methods that we used. And just a snapshot of the cartographic framework that we ended up coming up. So we thought that there's two parts of this work, where at first we have data that has been aggregated. People already have identified what to if you want to say drug resistance, how should you calculate it? So, so this would fall in post-positivism sort of correctness. So here we'll try to get antimalarial data from the resources that are, and here it was published resources and the routine data, summarize the evidence. And then when we are coming to making maps and evaluating maps, this goes beyond now, because here we have to use cartographic method. So there is a user's perspective. Me as a cartographer, I can project my feelings and knowledge into making maps. But then how do we minimize that? And then if I'm making a map for a certain user, how do I incorporate that feedback without um, um, sort of changing, sort of I have to interpret what they said and add it on the map. But then a health problem is, despite I'm being a cartographer, but I'm also an epidemiologist, in a way, not always um, users or malaria programs would like to see a health problem as we portray it. So at a point, then I become like an agent. I have to put the evidence as it is, despite it's not liked. So how do I sort of um, identify where I have to be an agent to explain the reality, but not jeopardizing or underplaying the feedback that I get from my audience. So, so I've put it here. I hope the slides will be shared for people who wish to read more. But so this is sort of, we had to come up with methods and iron out what we would be able to do. So I would sort of go straight to two sub-studies. One we did was to try to do improvement of routine surveillance data and systems for mapping resistance. And this was done here in South Africa, in Mpumalanga. And we had a team from University of Cape Town, the University of Oxford, the NICD, National Institute of Communicable Diseases, and then for South Africa. And we had malaria programs people. So this is the malaria program manager and the information officers. So there is a study called Smart Surveillance Study that was just rolled out in 2018. And the aim of the study was to do a real-time surveillance of drug resistance. So we rolled out this study to try to, so this is a simple schematic, sort of visual display. We know malaria happens in the community. And these people have places where they live, but they come to a facility to get tested. Eventually, for South Africa, malaria is a reported medical condition. So every case needs to be investigated. And this is what is recommended in most of the areas that are approaching elimination by WHO. So malaria case investigators go to patients' residential addresses. They collect GPS coordinates, addresses where these people live. But when these patients visit clinic, a blood spot is taken, they're getting antimalarial treatment, then they go back home. So these blood spots that were taken are sent to the laboratory, and drug profiling is done to know the kind of parasites that these patients had. And then, so smart surveillance, the aim was to be able to link individual patient data with, and their demographics with this malaria drug profile. And be able to sort of present it. First was to just sort of help the program to be able to identify, to have a sort of a warning system if we are developing resistance. But the second part is how do we represent this information? The best way was thought of using maps. And so these are the preliminary maps that we produced in 2018. So For what I just presented, if we want now to create a system and create maps for this program, how would we do it using the same methodology that we thought about? So simply, routine malaria data that we receive, it comes from malaria cases and laboratory data. We also get spatial data. So this spatial data, it means location data. It could be GPS coordinates, patients' addresses, clinic, where the clinic is situated. But apart from that, if we have to create a map like this one, there is a shapefile. So this shapefile is a type of data that is already available. You can either download it from open sort of website resources, free resources. You can also look for government resources to get this data. There are also international resources you can download. One thing that you'll note that despite we look at maps and we know that this is a map of, for example, this is Nkomazi in Mpumalanga, or a map of a country, let's say South Africa down here. There's a lot of alterations that can be done on the shapefile. And when you're coming to map a small area like in Nkomazi, which is like a dot right here, you start seeing these differences, which probably if you're looking at a country map, you wouldn't see. So this kind of spatial data that we would be looking at. And then, and it's not that if we're talking about mapping malaria, the same geographical unit from Komazi, people don't live everywhere. Malaria occurs where people live. A lot of this part here, if we have any South African friends or people who have visited South Africa, they know there's a Kruger National Park up here. And apart from that, there are big estates where you travel 2 kilometers of farmland, where only workers who come and go back to their residential addresses are situated. So if, for example, I put a dot on the farm, in reality, would I be helping a program? So these are kind of considerations that we would have to look at, and probably they wouldn't make sense if we're looking at a country map. And then we would have to curate now this spatial data to represent what is in Mpumalanga, and then be able to merge this spatial data with laboratory data. But the case data that comes from the information, for example, malaria case, malaria patients are attended at the healthcare facility. But the data that we end up having is the one that comes from the information system. Is it the data that represent the cases as they were attended at the healthcare facility, or does this data also have errors or any manipulations? So we had to sort of do a good data review to see that. But because of the logistic of drug resistance, every malaria case had to be linked to laboratory data, and we had to use a barcode. But these barcodes were to be captured by someone or to use a barcode scanner. And would they have transcriptional errors and things like that? So all those needed to be addressed before we link back to be able to create maps. So I'll give a snapshot. So this is from March to August 20. Oh, not 2028. This was supposed to be 2018. And what happened is the first map of cases location. So a little bit of context here. So the gray part, this is South Africa. This part here is Mozambique. Here we have Swaziland. And then Mpumalanga has it's bordering Mozambique on the eastern side, and it's also bordering Swaziland on the southern side. when we received these are supposed to be GPS coordinates. This is where a case investigator went to a patient's home to get information and uploaded it into the system. Unfortunately, a good number, like almost half of the coordinates, landed somewhere, say either in Mozambique, and further analysis we could find them. Some of them were in water bodies, and some were in Swaziland. So this is the reality. And so, for example, if we were to sort of use this data as it is to make maps, it would be a bit wrong. But then in reality is, where would this be coming from? Um, could it be either in healthcare facility, probably someone captured the wrong address or transcription errors in copying down, or because also we are working with migrant population, they might not say the right um, residential areas where they live. Some of them could be illegal migrants and they wouldn't want to be captured. So such things would affect that data. Then there are errors that could be due to location. In order to capture a coordinate, you have to use a device. Could it be due to device setting? Or could it be someone, after reading a device, copying the coordinate down, they got it wrong? Could it be due to the information system? Maybe a coordinate was okay, but once it's put into the information system, um, it could be changed and cause these errors. So we had to use the methods that we had put in place to evaluate this whole process and identify the problems. One of the things that we did, so this is a presentation for between March to August 2018. So I'll change that. And you'll see in the first 3 quarters of the time, we're still trying to address where these issues came from. And so we identified, so at first we went to the GPS devices to look to see if there are any issues. Case investigators were using 2080 GPS devices. 9 were these tiny eTrex 10 devices, and 19 were using Android tablets, or sometimes their phones. And all of them, almost all of them, had different units. This actually is a big problem. In a way, if the same coordinate captured in one unit is translated using another, they can give a range of 0 to 50 kilometers difference. So that definitely would place someone into water. But then they wouldn't explain why some would be in the northern hemisphere. So we ended up finding some where the mix of negative and positives into the system. And we also decided to take the coordinates that were just captured as they were and put them in the system. So despite the the 9 eTrex devices having different settings, at least a good number of the cases would still be in Mpumalanga. We ended up identifying all the 1900 devices. So watch out for Android devices if anybody's doing this type of data collection. Actually led to majority of the coordinates being far away from Mpumalanga. So, and part of the program was to be able to identify this problem and help the program to improve. So we created the SOPs, we conducted training, and we also provided system checks within the NMs, the capturing form, sort of to put a format so that someone wouldn't mix, where should I put negative or positive, or how many decimal digits have I to put. And then we also identified an error within the DHIS2 system. South Africa has been rolling out DHIS2 system since 2017, 2018, and some of them are programming errors within the DHIS2 system. And just a snapshot of how we worked on this. So these are the sort of Sankey diagrams to show how the data quality changed. I'll just bring you to this big sort of block here. So at first we had like I don't know, 10% eligible cases that were able to link molecular markers. So that means if we're going to make accurate maps, we would only use that, to almost having 51% of cases until February. So it has been a sort of a long activity, and part of this explain where exactly we were missing these coordinates. And I know I explained to you about the shapefile. We had sort of look for a shapefile that would be useful for the malaria program. If we would just present maps as they are, using any shapefile from the government. So here we obtained a shapefile from, so we used a standard shapefile. So we went to the municipal office, obtained a shapefile, and then this showed location of where people live. And then we added malaria cases on top of it. One thing that we realized, these shapefiles were actually outdated, were 2011. Sort of most of these settlements have grown bigger than what actually the official shapefile looks like. And we had to find another shapefile that would replicate the current population of Nkomazi. And it would evaluated using 24 case investigators. So malaria case investigators are people who go to these residential addresses to look for malaria cases. Some of them have worked for 24 years. A lot of them have worked for more than 10 years in the same area. So they know this geography quite well. And so we tried to evaluate, and eventually we came up with sort of a consensus shapefile that we would use to represent the cases. One thing that I can tell you, we first, the best agreed shapefile is the one that wouldn't use the geographical demarcations. So sort of this, so this is a sub-district. It has, these are the wards. Most of these wards, so for example, if we put a thematic map using wards demarcations, a lot of these places where people don't live, like a whole of this area would have a lot of cases. And this area is just a national park. So it seemed like if we use a population or settlement shapefile, it would be more easier for people to understand where malaria occurs. And the program said this could be more useful than just putting it per ward, cases per ward. And eventually, by implementing sort of the training workshop and doing evaluation while we were creating maps, we were able to, as you can see, this is the first few months of the start of the project. And then these were the last months of the project. So this is the data that came, raw data that came from the health information system. So majority of the errors that were within the system, we were able to troubleshoot them, and we evaluated to see the trend. Eventually, we actually would get bumps of good accuracy whenever it's a month after training, and then things would go a little bit down, then they would go back up. So there's a little bit of also sustainability in training and making sure people retain the knowledge within this. And I'll just keep that. And so what we did for optimization phase, so we just went back to what we wanted to do within our cartographic framework. Sort of we have summarized the evidence, we are creating maps, but then we would have to take these maps to users. So eventually have maps. So at first, these were the first maps that we developed. We had to sit with malaria programs and look at them, use the semi-structured tool to respond to a few questions. And then we would sit down and try to optimize their feedback to get a final map that would eventually share. And this map would contain sort of the cases as a background, and as well as the distribution of the molecular markers of the area. And here, these are the artemisinin Kelch markers, and we denoted them by red as sensitive. So sensitive, this means that that parasite hasn't yet developed resistance. They are also tolerant, resistant, and if they would be missing a result. And so that is one. And a short part is we tried to use the same framework to create sort of regional maps of drug resistance for Asia. And our process at first, as usual, you have to work with data. So we use the K13 surveyor database to be able to get the molecular markers that were already aggregated. But since it was a little bit updated, we had to update it with the newly published data. And since also in order to create maps, you need further geographical information, we would have to look at these studies and try to find exactly which are the study sites where these studies happened. So we conducted a systematic review, obtained the studies, then synthesized the studies to get the molecular markers and where they happened. So for example, one of the analysis that we did was to look at, are these study sites really in the areas where they have been reported? A few of the study sites you'll see, this is the geographical boundary for Thailand. And you'll see some of study sites that reported to be in Cambodia. If you put the either coordinates or just reported address, you'll find them in Thailand. And so a lot of inter-border, especially coordinates, ended up lying in a different place. And even within country, would find a lot of a few study sites that in the reports, their locations would actually land somewhere else on the map. And so it would be either to chop this out because, or go back to these authors, ask them about revision of their study sites so that they can be incorporated and put on the map. So for example, the one survey that I showed in the beginning had used some of these sort of study sites where you'd find that pin, and if you kind of investigated further, you'd see that that pin would land somewhere else. And that's how sort of an activity that would have improved, would actually improve the surveyor. Another thing is also, you find a lot of studies would report the catch markers. Some wouldn't report the location information. They'll just say in Myanmar or in Thailand, but you wouldn't know exactly where did this happen. And some wouldn't say whether it was a therapeutic efficacy study, was it a molecular surveyor, and how they analyze samples. So, sort of there are challenges that would happen in a routine system, but also we found some challenges that are almost similar that would happen even in just studies data that is being shared globally. So we did also usability assessment. So we used the same questionnaire that has 30 questions. We looked at how people would understand the map and eventually incorporate their feedback back to the cartographic process. So one thing that we can show that what actually changed much in Asia, in the regional maps, is first to get the study sites to be accurate. And second, some studies end up reporting similar samples in different studies. So it becomes very challenging to aggregate data, especially if similar scientists use the same data set to just change the analysis and they sort of publish. So such kind of things we're able to come around and address. Other things would be also, how would we present the map? Every time when we send a map back to the policymakers or malaria staff working in Southeast Asia to revise, it would have other sort of new feedback of what they want, and actually it has really changed how a map would look from the beginning, how you start the second map and then the third map. But also the molecular markers are increasing, so some more countries are coming into play, and some of them we haven't been able to assess that. So this is, I hope, is my last slide. So there's a lot of challenges within using this method. At first, a lot of data is not used. So for example, if you start with the published studies, a lot of data is not uniform. So we had to create a tool, and we have published it in one. So to sort of suggest to researchers, ask them if they can somehow try to have minimal information so that these kind of studies can be pulled together. It's a very important practice, because if we are going to analyze global transmission of malaria, sort of resistant malaria parasite, we have to have a uniform language. And if this data doesn't talk to each other, these data sets, it means we can't be able to do it as we are supposed to. And probably supply data formats for that, studies that will require maps. A lot of studies don't put a lot of emphasis on location information, which end up in a lot of maps. So also probably having a location validation protocol. If one is entering data, for example, a data set like the One Surveyor, if they enter an address, and if that address ends up in a different country, they can definitely know that this is the wrong address. So sort of having sort of system. Another thing that can be done for routine system that we tried to implement was to change the form that is being used, and also to add checks on DHIS2, which is the routine system. And also create sort of repository of just locations or study sites. So you'll find a lot of these studies, for example, the ones that have been reported in Africa, they are coming from sentinel sites that are already known. So if we had a database of where these sentinel sites are, we can be able to sort of cross-validate where this information comes from. So eventually, that's what I had in mind to present. I don't know if it fit the context of the presentations, but that's mixed methods in my study, and that's how I've applied it. I've applied them. Challenges is a lot of work wasn't expected, especially on data. That took almost 70% of the time. It's difficult to do feedback, especially, malaria programs, people are very busy, have a lot of work. And sort of if you to sit with them for feedback, and then design with them activities that would you'd bring back to the program staff to work on, it takes a lot of time. So time can be quite an issue. And then another challenge, it needs a lot of being tech savvy, as well as analysis, sort of to learn different languages, so that you can be able to operate. It's easier when you have a big team, but then if you don't have a team as a PhD student, you kind of have to develop the skills yourself. And then the other challenge is publication. Let's say a lot of this work cuts across, it's sort of iterative. And a lot of journals, when you sort of communicating with the reviewers, it's in between trying to justify why you did what, and you can't publish your manuscripts together. So it also can be challenging. For example, we have decided to publish quantitative papers separately and qualitative papers separately. So those kind of things sort of can be challenging.